Hello, you are welcome back to Cell and Tissue Organization course. Under Section 3 of Unit 6, we will look at staining, which is very essential in the preparation of tissues for study. We will begin with the need for staining and look at some common stain types and then end with the methods of staining. By the end of this session, you should be able to explain the need for staining tissues and list some of the common stains used in histology, and also describe the effects of some common stains, and also describe the different methods or types of staining. Beginning with staining, staining involves the use of artificial coloration of a substance, normally referred to as dyes, and these substances help to facilitate examination of the tissues. It could also be microorganisms and other cells under the microscope. The purpose of staining ensures that cells are primarily stained to enhance visualization of the cell or certain cellular components under a microscope could be also visualized. It also highlights certain metabolic processes or helps to differentiate between live and dead cells in a sample. The principle of histological staining relies on the treatment of tissue sections with dyes in solution. So for most cells and cellular elements are virtually transparent, and because of that it is difficult to distinguish individual cells and cellular structures without staining the cells in some way before viewing them. That is why there is the need to stain your tissues or cells or microorganisms in order to view them under the microscope. Now let's have a look at stains types. There are several types of stain and each can be used for a particular purpose. And two major ones we will be looking at is hematoxylin and eosin, which are usually abbreviated as H and E. These are commonly used histological stains. The hematoxylin is a basic dye and they therefore tend to bind to acidic structures and stain such structures blue. The most distinctive acid in cells is the nuclear DNA, which consequently the nuclei appear blue when stained with hematoxylin. The structures which are acidic are said to be basophilic, that is, they are able to attract basic stains. For the other type, which is eosin, this particular dye is acidic and they therefore stain basic structures red in color. The cytoplasm of most cells is slightly basic, and because of that, they stain pink, and they are said to be such structures, or the cytoplasm is said to be acidophilic because it also attracts basic structures. A knowledge of these properties of the stain type used will allow you to interpret a preparation made with an unknown stain. And so the nucleus is always acidic and therefore defines the basic stain. For a staining procedure, the traditional or conventional method involves the use of dyes that selectively binds to different structures within the cell. However, recently, techniques that require the use of antibodies have also been developed, which can stain individual molecules. The two main types of or methods of staining cells are known. The first one is progressive staining, which involves or is a procedure in which the stain is applied until the desired intensity of tissue coloration is attained. And this involves leaving the tissue in the stain until, for instance, the nuclei, as an example, are deeply stained and the cytoplasm is faintly stained. And for this procedure, it is good to dilute the stain in advance to reduce the rate of staining and the risk of overstaining. Overstaining would reduce the contrast due to deep staining of the cytoplasm. This is one drawback of this particular method. The second method of staining is retrogressive staining or regressive staining. And this type of staining is deliberate or involves deliberate overstaining followed by destaining, resulting in differentiation. And the best results are obtained when overstaining is prolonged. This method relies on differential rates of stain extraction. For instance, acid alcohol removes hematoxylin more rapidly from the cytoplasm than the nuclei. An advantage of this particular type of staining is that it gives sharper differentiation than progressive staining. It is possible to obtain deeply stained nuclei surrounded by colorless cytoplasm. Under Section 3 of Unit 6, we've considered the need to stain tissues before examination under a microscope. We also studied some of the stains used and how they are used.